Hello dear learners, today in this video we are going to discuss the topic incomplete dominance from the lesson Principles of Inheritance and Variation. So students, before we understand what is incomplete dominance, let us rediscuss about the laws of inheritance that was proposed by Mendel. So he has given two laws based on his monohybrid cross, that is law of dominance and law of segregation. So according to him, that the laws of inheritance states that the characters are controlled by a discrete units, and these units are known as factors. And secondly, factors always occurs in pair, and number third, the factors can either be similar or dissimilar. So this is what he has to say in his law of dominance. Whereas in law of segregation, according to Mendel, it states that out of the pair or the factor, they segregate or separate during the time of gamete formation, and they re-express themselves. I mean, the recessive trait, which could not express every generation, they can re-express itself when they come together, as there is no mixing, right? According to him, out of the pair, they do not influence each other. As a result, they are separated independently, or they are segregated during the time of gamete formation. So this was what about his idea from his monohybrid cross. But if the same steps or the process is used in order to find or to study the inheritance pattern in other plants, or if we consider some other characteristics, the result somewhat we find it came out to be different. I mean, it is deviated from what Mendel has proposed. So according to Mendel, the result of monohybrid cross come out to be 3:1. That is the phenotypic ratio, whereas the genotypic ratio is 1:2:1, right? But here in incomplete dominance, say for example the plant Snapdragon, which bears different flower. So if we consider the plant with flower red and the other plant with flower white color, if we cross, the result is expected to come in red, as if we follow, or if it follows the Mendel's monohybrid cross. But what we find here is that it is completely different. Instead of red, it comes out to be pink in color, right? So let us see how it can come. Let us see here with the help of a cross. I'm going to show the red flower with this red color in when it is crossed with a flower with white color. So I am using blue here to denote white flower. Suppose this is white flower and has the genotype small r, and this is red flower with the genotype capital R and capital R. So when they are crossed in the first generation, F1 generation, so this is P, parental. Okay, so P stands for parental generation. So in F1 generation, if we are supposed to get red color, but it is not so. The result changes from red to pink, means it's neither red nor white. Here red cannot express itself completely, or red color is not a dominant trait here, as it cannot dominate completely on white color. So as a result, the color turn out to be pink in color. So we are supposed to get a red colored flower, whereas it is not so. Instead of red, it comes out to be pink in color. So this flower I am showing with red and blue, so to denote you the color pink, right? So what will be its genotype? As according to Mendel, the law of segregation states that the factors separates or they segregate. So as they are homozygous, so it will be same type or same gamete each of the plant will provide. So capital R here and small r here. So it will be genotype will be capital R and small r. So this is F1 generation. Right? Now, by self-crossing F1. Now, by self-crossing F1 generation, what we get? Suppose F1 generation we cross between two pink flowers. Suppose as I am showing pink with red and blue mixed color, right? Red and blue. So when they are crossed, so result we get is in the ratio of 1:2:1. That is, one completely red in color, one is completely red. Then there are two pink, right? There are two pink flowers and one white completely. So the result here, the phenotypic as well as the genotypic characters or the ratio that we obtain is, suppose genotypic ratio we obtain 1:2:1. That is, one homozygous red, two heterozygous pink color, then one homozygous white. Whereas phenotype also same. Phenotypic character what we get? One red is to two pink is to one white. Right? So this is the phenotypic feature. So this is how we can find the changes or the differences that we obtain in a monohybrid cross. Monohybrid cross, it is expected to be red in color, as red color is a dominant trait. So in F1 progeny or in F1 generation, all the flowers should be red in color, but it is not so. Here the color red changes to pink, as because here the red could not express itself completely, or the red did not dominate the white color completely. As a result, the color came out to be in an intermediate color, that is pink color. So this phenomena is called as incomplete dominance. So this is the case, the example of a flower called Snapdragon, right? Where both the parents are, one is red and white, but the F1 progeny come out to be pink in color. But in the next generation, after self-crossing the F1 generation, both the parents can be retrieved. That is, one red and one white, whereas two has got an intermediate color, that is pink. So this is about the incomplete dominance. So students, let us understand about the concept of dominance. What exactly is dominance, right? So as Mendel already has said, and let us also recall about what Mendel has given in his laws of inheritance, that characters are controlled by a discrete unit which he termed as factors, and factors are in pairs, and pairs can be similar or dissimilar. So by now we have already understood that these factors are actually the genes that control the characters, or the phenotypic expression is due to the gene, right? And this gene always occurs in pairs, or the gene always have a set. I mean, it has another copy or a set that expresses for the same phenotypic expression. So whenever there will be some changes in the gene, and definitely these changes in the gene is going to affect or is going to change its phenotypic expression, right? Say for example a gene that synthesize a certain enzyme, right? So this can either be, there can be three different cases. Suppose the gene, one set of the gene, the set of the gene can produce the normal enzyme, right? The normal enzyme, and in the second case, the set of the gene cannot, or it synthesizes a non-functional protein, or number third case, there is no production of enzyme at all. So due to modification. So ultimately this modification in the gene will definitely going to affect to its phenotypic expression. So this is how it will deviate from its character, right? It will change its character as there is change in the gene. So more or less the unmodified gene, it does not affect much to the phenotypic expression, and as a result it will be considered as a dominant trait, and it will be able to express in the next generation in its phenotypic expression. Whereas in other two cases, like where there is no expression of any enzyme, or the enzyme is non-functional, so in that case such trait will be a recessive trait, right? So this is what is about dominance and recessiveness, or dominant character and a recessive character, which is due to the gene, whether the gene got modified or not. If there is some modification in the gene, so definitely it is going to affect or going to change in its phenotypic expression, right? So that is it. I hope you have my explanation will help you. So thank you so much and keep learning. Bye bye.